Hello and welcome to this Bioprocess Insights webinar. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Madhu Raghunathan from GE.
All right, thank you so much, Leah. Welcome everybody to this webinar. Before we start, just a couple of quick words about myself. I am the product strategy leader for downstream bioprocess equipment at GE Life Sciences. I've been with the company for about 6 years now. My professional experience spans about 15 years, almost all of it in product development, technology commercialization, product management, and marketing strategy. So really looking forward to going through this presentation with you today. I'm going to start off by talking a little bit about a couple of things. We will start with the evolution of the biopharmaceutical market, especially over the last decade. We will then talk about how does that actually set the stage for what is known as downstream process intensification. We will talk about what that is, why is it important, and how do you actually go ahead implementing process intensification. We will then talk about some applications and case studies. And finally, we will wrap the seminar up with some summary slides. Let's start with the beginning. So we all know that we've had a really good couple of decades in terms of the development of biological drugs. As you can see from the chart, it's not only limited to the monoclonal antibody class, but also spans some of the other classes of biological drugs like vaccines, recombinant proteins, plasma proteins, and so on. Along with the development, there's also been a few other market trends or market dynamics that have kind of taken place over the past several years. It's kind of been captured in this slide. What we're starting to see is fewer blockbusters when it comes to the biological drugs. We're seeing a lot more competition, especially around biosimilars, a lot more cost pressure, and also localized manufacturing. If you kind of start to put all of these trends together, what emerges is basically a shift or a transformation towards more multiple product and flexible manufacturing facilities, a shift towards newer therapy areas like antibody drug conjugates. There's also a greater demand on productivity, plant utilization, and a movement towards fewer variability and fewer batch failures and so on. What we're also seeing is a cultural shift, a mindset shift from what previously used to be known as the process being the product to a situation or a paradigm where we're willing to let the process be adaptive towards the inputs. And the intention here is to have a more process-based understanding, like QBD, for example, and ensure that the quality cost of manufacturing of biological drugs is actually reduced as much as possible. If you start to kind of piece all of these trends together, what we see is that there's a greater openness towards adoption of newer technologies. What we need to kind of keep in the back of our minds is that not all new technologies are created equal. Right? First of all, we need to kind of ensure that there is a performance and economic benefit when looking at adopting new technologies, and also some other aspects, looking at things from a regulatory perspective, scale-up perspective, the comfort level of an organization with these technologies before you kind of start implementing these technologies. The last thing we're going to talk about from a market development perspective is the increasing efficiency from an upstream perspective. Where previously we used to see titers of 1 gram a liter, we've started to see, even on commercial processes, titers of about 4 or 5 grams a liter, largely due to better expression systems, cell culture and process control strategies, and also improvements in upstream biopharmaceutical equipment. right? So this is basically a quick introduction in terms of what's been going on. And what I'm going to do now is tie all of this together and then move to the meat of the talk, which is to kind of look at how does this relate to downstream processes and what can you do in order to intensify downstream processes to keep up with all of these developments. Right? We're going to talk about four primary things. The first thing we're going to talk about is we're going to talk about chromatography unit operations and implementing that using automated packing or ready-to-use columns. The second thing we're going to talk about is creating closed or functionally closed systems as a way of increasing the efficiency of downstream processing. We're going to talk about single-use technologies, and we're also going to talk about continuous and connected processing. And we're going to tie all of this together with a common thread, which is automation. Right? So this is kind of how I see process intensification as a combination of all of these different technologies or solutions which can be applied independently, which can be applied together as a way of intensifying downstream process operations and therefore kind of enabling processes to keep up with some of the changing market dynamics that we kind of talked about in the beginning of the presentation. Right? So let's jump right in. Let's talk about the first thing on our agenda, which is to kind of talk about how do you intensify chromatography unit operations. Right? Let's start with the basics. When we talk about chromatographic purification, the key thing to kind of keep in the back of our minds is you want to ensure that there's an optimally packed bed. Right? Why is that important? Having an optimally packed bed, and not just the bed being optimally packed, but ensuring that the bed is also stable over multiple cycles is important because it has a huge impact on the performance, on purification, on yield of the product that's being purified. Right? When you actually end up packing a column, there's certain things to keep in mind. How do you ensure the repeatability of packing? How do we minimize or even eliminate bioburden risks? And how do we kind of minimize the time that's actually being spent on packing, testing, repacking, and QC validation activities? Right? Historically, the evolution that we've gone through when it comes to clean and reuse columns is that we started off with the methodology known as flow packing. Then we moved on into pack and place columns, either with manual nozzles or more off late with automated nozzles. The challenge in doing that is that there is a lot of manual steps that have to be undertaken in order to get to the final result of having a packed bed that is both stable and optimally packed. Right? This is just a very small snapshot of the number of steps that one has to go through in order to get to that end result. And even here, there's a lot of things that can go wrong. Right? So if that's the case, then what's the solution for us in order to intensify our chromatography unit operations? The most recent development as far as packing beds has been the movement or transformation towards using automated columns for achieving this result. Right? Why an automated column? An automated column is really helpful in order to address some of the pain points that we talked about. A lot of the times these automated columns employ axial compression technologies, which are more suited for modern resins. They also come with pre-tested and pre-verified methods, especially if these are intelligent columns. Right? The upshot of that is that there's no need to develop new packing methods. There is a reduced burden on operator training. A lot of the times just a single operator is enough to be able to pack a column. And from a sanitary perspective, there is a significantly reduced risk of bioburden, and the maintenance operations are also simplified. Right? Sounds like a good deal, but how do we know that this actually works in practice? Right? So what we're going to talk about now is a real-life case study where we implemented an automated column into the chromatographic unit operations workflow. And as you can see, what we see here is that there's almost a 50% reduction in terms of the amount of time that you have to spend on packing, unpacking, and maintaining columns when using, in this instance, the Axichrome column as opposed to a manual pack-and-place column. Right? So this is exactly what process intensification is about in a nutshell. How do you ensure, or how do you put in practice some of these technologies and some of these solutions that allows you to lean out your process, that allows for a faster cycle time, that allows better replicability, and that takes out some of the attendant errors that kind of come into the picture when using manual processes or manual workflows. Right? So this is one really good example to kind of get us started. The next thing that we're going to talk about is, column packing is one critical aspect of chromatography unit operations, but it's not the only aspect. Right? If you kind of take a step back and look at the bigger picture, that bigger picture starts from the time the resin arrives at the facility and going through the workflow of removing the supernatant from the resin container, filling in the packing buffer, homogenizing the slurry, measuring the slurry concentration, transferring that slurry either via a slurry vessel or a slurry tank into the column, and then getting to the point of having a packed bed and making sure that the bed is well packed by running a HETP test. Right? A lot of the operations here, once again, currently are run in a manual way. So the benefit in kind of automating this and running this as a closed system or a functionally closed system involving aseptic transfer of the liquid or the slurry through the entire workflow, the benefits are significant. Right? It ties down to kind of the things that we've been talking about in terms of increased process outcomes, but also a significantly reduced bioburden risk or cross-contamination risk. Right? So far we've been talking about chromatography unit operations from a clean and reuse perspective. There's a completely different perspective that we can take here, which is to kind of utilize pre-packed columns in order to do the purification step. Right? This is an example where we looked at the workflow comparison between clean and reuse columns and pre-packed columns, and the end results are really impressive. As you can see here, we've kind of eliminated 7 of the 11 common steps associated with column packing when you make the shift towards pre-packed columns. Right? So that being the case, why can't we always use pre-packed columns? Right? Pre-packed columns are not necessarily single-use columns. They're more considered as campaign-use columns, and the decision factor comes down to a few critical parameters. The first thing we need to think about is the production volume or production scale. We need to think about the manufacturing frequency. We need to think about the nature of manufacturing operations, whether we're producing a single product off of that process train, or are we producing multiple products. What is the internal know-how and knowledge base when it comes to cleaning, validation, and column packing? And what are some of the considerations around CAPEX, OPEX, and replicability expectations? Right? So once you kind of have these things in the back of your mind, then it becomes a really easy decision in working with your vendor in deciding, should you go down the path of using clean and reuse columns, or should you go down the path of using ready-to-process or pre-packed columns? Right? When we're talking about pre-packed columns, we don't have to necessarily limit us only to columns. We can kind of broaden the envelope and then talk about single-use technologies as a whole. A lot of us know the benefits of using single-use technology, the key benefit being the minimization of risk, cross-contamination risk, and the reduction in time that's spent on cleaning, validation, and sanitization. Okay? The upshot of this is a faster turnaround time or a shortened cycle time between batches or products, and increase in utilization of equipment or uptime. What is also really important when going down this path is to kind of keep in the back of our minds that you need to have a robust supply chain behind your operations in order to ensure that single-use technologies are being kind of put in place in a plug-and-play fashion. It doesn't really help when there are constant questions around bag integrity, or there are questions around extractables and leachables that are kind of coming out from these single-use products, or if the vendor cannot actually deliver these single-use products in a timely manner. Right? Therefore, thinking about security of supply when utilizing or implementing single-use technologies is really critical, as is some of the other things that we've talked about from a supply chain perspective. Right? Jumping on in terms of results or outcomes, like we kind of looked at when we were talking about column packing. When you have a completely single-use workflow, in this instance having a chromatography system with a single-use flow path and a pre-packed ready-to-process columns, you can see that there's a significant reduction in the setup time associated with getting to the point of purifying your protein. In this specific example, there is a 10-fold reduction in the setup time when using a chromatographic system with a single-use flow path and a pre-packed column. Right? Hugely beneficial because you can use that time. There's an opportunity cost here, and you can use that time to do other things. You can use this time to kind of get more products out of the pipeline. So there's a lot of benefits in terms of taking this path, but we have to, like we kind of just discussed a slide earlier, we have to kind of carefully think this decision through in terms of when we want to do this and when it's suitable to be to kind of go down this path. Right? All right, changing tracks a little bit. So far we've been talking about chromatographic unit operations. What I'm going to talk about next is around two connected topics around continuous processing and something called a straight-through processing. Right? A lot of us are familiar with what continuous processing is, and for the purposes of this talk, I'm going to focus on continuous chromatography. In this instance, periodic countercurrent chromatography. Why is that important? Why is there a certain interest in terms of continuous processing and continuous chromatography? If you kind of take a step back and then do the comparison between batch and continuous chromatography, some of the key benefits when moving towards continuous chromatography, the first key thing is, depending on where you start and what your ambition level is, there can be a significant gain in terms of the capacity utilization of the chromatographic resin. The way in which batch processes are run, the batch capacity of chromatographic resins may not be fully utilized, and that's a big benefit when making the shift from batch to continuous chromatography. There's also a reduction in equipment size due to what we just talked about, due to the fact that you're able to utilize more of the resin capacity, and therefore you can go down in column size and equipment size when making the shift from batch to continuous. Continuous chromatography also enables continuous bioprocessing. The integration of bioprocessing starting from, for example, a perfusion bioreactor all the way down to the concentration step. And this is especially useful when dealing with products that have stability issues, and therefore continuous chromatography, continuous processing allows for an improvement in product quality and also allows for better robustness and control of the entire process due to the steady state nature of the operation. Right? So in terms of benefits, here is a chart that kind of compares a few aspects of continuous chromatography as opposed to batch chromatography. And we're looking at two separate aspects here. One is around the capital expenditure, and the other is around operating expenditure. Right? The details aren't nearly as important on this slide as getting to the point that depending on the starting point and depending on what the ambition level is, there's going to be a lot of different benefits around continuous chromatography. There's also going to be a few areas where continuous chromatography isn't going to be nearly as attractive as batch chromatography. So we have to keep that in mind, and having that kind of thought process and saying, where am I gaining, where am I not gaining so much, and going through the thought exercise is really valuable in terms of understanding the practical implications of implementing continuous chromatography. Right? In terms of a related topic, which is straight-through processing, which is not purely about continuous chromatography, it's a way of ensuring that the elution pool from one step, the purification step, for example, is directly loaded into the next step, a polishing step maybe, without having to have a hold-up tank or having to do some intermediate processing in between those two steps. Right? The benefit of doing this by no means is a completely novel concept, but the benefit of doing this is that there is going to be, due to the inline adjustment, there's going to be a reduction in the total footprint of equipment, and there's going to be an overall reduction in the cycle time of your downstream processing. Right? A related topic to straight-through processing, especially as it relates to buffers, formulating of buffers, handling buffers, and so on, is around something called as inline conditioning. Right? Inline conditioning is just a fancy term for saying how do you actually automate and how do you make the process of buffer preparation and buffer handling more intelligent and more automated. A lot of us are familiar with the pain points associated with buffer formulation and buffer handling. Especially as you go up in scale, buffer handling and buffer preparation necessitates large tank farms, preparation of these buffers ahead of time, moving these buffers through facilities in a cumbersome manner. Right? So how do you make that process more intensified? How do you make that process more intelligent? Inline conditioning is a way of solving that problem. Right. What you can do is, what inline conditioning basically means is that you prepare buffers or you formulate buffers on the fly from 4 basic stock solutions, which is basically acids, acid, base, salt, and Wi-Fi. Right. Depending on the recipe that you have in place, you're going to be able to get to your final buffer right away. And the inline conditioning logic or algorithm actually is intelligent enough to kind of accommodate for variations of your input parameters and still generate the output that you want. Right? So how does this actually work in practice? What we can actually see here is an example or a case study of where inline conditioning was implemented successfully and leading to a 10-fold reduction in buffer volume and almost a third reduction in the footprint of tanks and systems that were previously used to prepare buffers manually. Right? Just to kind of make things a little bit more clearer, there's 2 different modes, if you will, or methodologies of creating buffers dynamically and on the fly. The simpler methodology is what is known as inline dilution, where you take concentrated buffers and dilute them just in time. The more intelligent or the more superior methodology is inline conditioning, which is inline formulation of stock solutions, like we talked about, from the stock, from the basic components. The advantage over inline dilution is that this process or this methodology is dynamic, and therefore it takes into account some of the variations of your input solutions. Right? So having talked about chromatography unit operations, having talked about continuous connected processing and also inline conditioning, and we also touched upon single-use technologies, we're going to try and wrap everything under the umbrella of automation. Right? So here's a guy who probably knows a thing or two about automation, and we kind of have to trust what he says here. How does this kind of impact us from a biopharmaceutical or a biomanufacturing perspective? Automation doesn't have to be either an on or an off switch. Right? A lot of us tend to think of automation as 2 discrete worlds. Either you're in an automated world or you're not. It doesn't have to be that way. As you can see here, the starting point of this journey is being in a situation or having a workflow that's not automated. And then from there onwards, what we can do is to kind of move towards a setup where you have disconnected islands of automation. These islands of automation could be running on the same automation platform. They could be running different automation platforms. What we can then do is to kind of integrate the data that's coming out of these islands of automation and integrate them or kind of gather the data into a backend or a backbone for further analysis, analytics, and so on. And moving on from that point, what we can do is to kind of have an integrated UI that runs all of your automation or all of your workflows in an integrated fashion. Right? The next step in this journey is to kind of have combined unit operations where you have a single recipe that's kind of running all of your different unit operations. And finally, the nirvana or the end goal, as it relates to automation, is to have a integrated facility that's being run from a command center of a recipe, and all of your data acquisition, data analytics is all integrated into a single back end. You're able to run your process in a dynamic fashion based off of QBD or process analytical technologies. You're able to do real-time product release. You're able to dynamically adapt the process in terms of the variations of your inputs. So this is the end goal, but again, we don't have to kind of make things complicated and say, I need to get there and I need to get there in a single step. It doesn't have to be that way. You can think of this as a stepwise transformation where you start off in a world where things are minimally automated or not automated at all, and then kind of end up in the automation nirvana. Right? So that's basically what we wanted to cover in terms of the different process intensification techniques. What I'm going to do is kind of tie everything together and then wrap things up by saying the things that we talked about, right? Improved column handling, elimination of cleaning and validation through single-use technologies, simplified buffer preparation using inline conditioning, increased productivity through connected and continuous processes, and increased system integration via automation. All of this ties back into what we talked about initially in terms of the market dynamics and market trends. And all of these things, either together or independently, have an impact on the amount of flexibility that you can achieve on your manufacturing process, the amount of footprint reduction, reduction in downtime and cycle time. And why this is important is, like we talked about right at the beginning of the talk, all of this ties back into increased cost and process efficiency, which is exactly why we kind of how we set the stage, and we kind of started talking about why this is important for us. Okay? So all of these different techniques tie back into improved cost and process efficiency, and that is why we must kind of keep this in the back of our mind or consider them as we go through this journey of process intensification, especially downstream process intensification. Okay? So that brings me to the end of the talk. Thank you so much for your time, and now let's open up the stage for any questions that you guys might have.
Thank you, Madhu. And thank you everyone for joining us today. So we're now going to start your questions. Just as a reminder, if you go ahead and type them in the chat window, we will get to answering them. So the first question is, what is the biggest challenge in implementing continuous processing in downstream bioprocesses?
Yeah, I mean, I would say there's a couple of things that we think about in terms of challenges. The first thing is the complexity of continuous processing and continuous manufacturing, which kind of puts thoughts in the minds of people, and which kind of makes the switch a little bit unattractive. A few of the other things in terms of challenges is there's also like a hesitation in the minds of people, especially around the integration of upstream and downstream process and having a fully integrated, a fully continuous process, especially as it relates to contamination. Right? The risk is that when you have such a continuous process, what happens when there's contamination, and how far do you have to go back in order to ensure, quote unquote, your batch is actually meeting the specifications that it needs to meet. Some of the other considerations are that the development process itself is a little bit more complex. You have to think about the fit into an existing facility, which might mostly be run in a batch fashion. The organizational understanding and the organizational maturity around continuous processing, either from a step perspective or from a start to finish perspective. Also from a technology perspective, I think there's a few things that we have to do in order to make sure that we have robust sensors for making sure product quality is where it needs to be. And also, for example, when you're running a continuous process over a really long time, especially running it for 30, 60, 90 days, how do you make sure that your process continues to remain sterile, right? So I would say those are some of the things, constraints or challenges that we have to think about when we start implementing continuous processing as part of our workflows.
Great, thank you. Another question is, do you know if there is a commercial process that uses continuous or straight-through method?
Yeah, in terms of numbers, I mean, we did a little bit of work trying to understand how many commercial processes are using continuous processing. From an upstream perspective, I think what we came up with was about, if I remember right, about 18 or 20 processes using perfusion bioreactors or perfusion culture as a way of doing their upstream processing. On the downstream side, we actually didn't find any commercially approved process implementing continuous processing. This probably has, it's probably related to kind of the previous answer, but it's also probably related to the fact that perfusion technology has been around for a long time, and I think there's familiarity with it slightly more than than the familiarity associated with continuous manufacturing on the downstream side.
Okay. How can intelligent columns handle significantly different resins, like ceramic-based resins or agarose-based?
Yeah, I would say there's two aspects here when it comes to intelligent columns, right? One is the methodology itself in terms of how these columns get to the point of having a packed bed. And the other aspect is around the way in which the intelligence is built into the column, right? Which is the pre-packed methods. The thing with the methodology is when you use the axial compression methodology, more often than not, it allows for you to be able to pack some of the modern resins, the resins which are more rigid. So that's a really good starting point, and that's a constraint when you're using some of the other packing methodologies. Like, for example, if you're using the pack and place methodology, there's actually a restriction in terms of how much or till what diameter and till what bed height you can actually pack the modern resins or the rigid resins. In terms of the specifically in terms of your question in terms of ceramic resins, I would say it depends. Depending on the type of resin that's being packed, the axial compression methodology should help there. And the other thing is, as long as the intelligent packing method recipe is already there, or if it's not there, it can be quickly developed and put in place, and therefore you can use that as a way of packing the specific resin that you're talking about.
Okay, and we have a couple questions about the study and where you got the data from. Was it a GMP facility? Was it a GE manufacturing facility? Can you share a little more about that?
So, we've kind of talked about a couple of different case studies. As far as I know, almost all of them are in GMP settings, and all of them have been together with external collaborations with customers, with clients. So yeah, they were in a manufacturing scale, and they were for the most part in GMP settings.
Okay. And what is the largest pre-packed column available?
I don't know the answer off the top of my head. I think we go up to 40 centimeter diameter right now, and we might even have something larger than that, but it's at that scale.
Okay. And what is the role of process analytical technology tools in continuous processing?
Yeah, the thing with process analytical technology, especially around continuous processing, is that you want to make sure that I would kind of take a step back and say the burden of making sure that your process is under control and your quality specifications are met are higher on a continuous process than on a batch process. And therefore, it's really important that you have the right type of process analytical tools, and the expectations when it comes to process analytical technology are higher. The reason for that is both from a contamination perspective, but also when you're running a continuous process over an extended period of time, you want to make sure that nothing untoward happens, or if it happens, you can pick it up right away. And if there are actually some minor variations on your input parameters, your process is able to dynamically adjust to some of those variations. So to that extent, I would say process analytical technology has a significantly greater role to play when it comes to continuous processing over batch processing.
Okay, and just two more questions, just for the sake of time to the audience. Thanks for sticking with us for so long. Is the minimum process size where continuous processing what is the minimum process size where continuous processing becomes most cost efficient?
Yeah, it's actually it's a good question, and if when we started looking into this, it was a little bit counterintuitive. Continuous processing actually makes more sense on smaller scale than it does on larger commercial processes. The reason for that is when you're doing, for example, clinical manufacturing or low frequency manufacturing, you aren't nearly utilizing the full life cycle of the resin, and therefore, being kind of putting in place, I'm specifically talking about continuous chromatography here. Putting in place, continuous chromatography is really beneficial at that scale. And as you start to go up in scale, as you start to look at commercial manufacturing, where your process is already extremely optimized as a way of keeping your efficiency high and your costs down, continuous chromatography isn't nearly as beneficial, right? But then, it also depends on a lot of the other starting points in terms of what does your manufacturing frequency look like scale, like we talked about, what is your flexibility requirements. So you have to keep all of these into account and then take a decision. I don't think I have a magic answer that says, here is the scale where continuous processing becomes more cost effective. But you got to kind of think about all of these things and then say, what type of processing makes more sense for me? But from a continuous chromatography perspective, like we talked about, the earlier scales are definitely where you see more benefits as opposed to going up in scale and ending up in large commercial scale manufacturing.
Okay. And the last question, and if we didn't get to your question, they will be passed along to our speaker with your email. So the last question is, have the intelligent columns increased the resin life cycle? Have
the intelligent columns increased the resin life cycle? It's a good question. I'm kind of tending to say probably. So what the intelligent columns do is ensure that you actually get to an optimally packed bed right away, and you don't have to. You're not going to have failures around your column packing, right? So I think to that extent there will be an increase in the life cycle of the resin. The reason being that, let's say that your typical batch failure rate or your typical packing failure rate, if you kind of take that into account, and if you have to pack and repack that column quite frequently, your quality of your resin is going to go down. So as opposed to that, when you don't have to, when you can get your packing first time right, and when you have an optimally packed and a bed that's actually stable over the number of cycles of operation that you want, that does have an impact on the life cycle of the resin, right? So I would say it definitely does have an impact on the life cycle of the resin. But it also depends on, what is your starting point? How often do what is your packing failure rate? How often do you have to pack and repack the column from a packing frequency perspective? What is the type of resin that you're packing? Is it does it lend itself better to kind of being repacked versus resins which don't lend themselves in a very good way to being repacked? So I would say all of those things play a factor, but if I have to kind of pick one side or the other, I would say that intelligent columns would definitely have a positive impact in extending the life cycle of resins.
Okay, well, thank you very much, Madhu, and thank you to our audience for joining us.